Yeah, good morning, everyone. I'd like to welcome you to today's Single Cell Omics Germany event, where we will learn about the work of Adrián Granada. And before we turn to his work, we would like to also announce the next lecture that will be on November the 19th, so in 2 weeks. Here, Michael Ratz will present his work on next generation lineage tracing in the mouse brain. And as always, so if you have questions, and we encourage questions and active participation, please write them in the chat or raise your hand. And yeah, with that, I would also like to introduce Adrián. So Adrián studied biophysics or studies physics at the University of Buenos Aires, and then did his PhD in biophysics here in Berlin at the Humboldt University in the group of Hans-Peter Herzl, studying the circadian system. He then joined for his postdoctoral work the group of Galit Lahav at the Harvard Medical School, and also worked as an independent research fellow at the IRI Life Sciences hosted by the lab of Niels Blütgen and Simone Reber. So he investigated during his career how the proliferation status and how the cell cycle affects the response to chemotherapy. Then in last spring, he founded his own group at the Charité Comprehensive Cancer Center, and combines in his group wet lab work as well as dry lab work, so long-term live cell, single cell imaging, as well as mathematical modeling to study how cells make decisions in the presence of DNA damage. And Adrián, we are very happy to listen now to your work. And yeah, the stage is yours.
Well, thanks for the introduction. And I will now share the screen. Can you see that? Yeah, cool. All right. So, move this. I'm not sure if you see this. So today we'll talk about our work on the effect of proliferation and cell cycle phase in the response to chemotherapy. And Stephanie said, I started my lab last year in the Comprehensive Cancer Center. We are part of the translational unit, and this is based on Charité here in Berlin. And the overarching question in my lab is to understand the sources of response heterogeneity in the context of anti-cancer drug treatment. And many of the drug treatments in the context of cancer, for many of these drugs, the most damaging effect that they have into the cells is the damage to the DNA. And when cells receive DNA damage, they have three outcomes long-term cell fates. One is that the cells can recover from this damage and continue proliferating. Otherwise, the cells can die or arrest. And broadly speaking, we can separate in two main sources of heterogeneity that can drive cells to one other outcome. There's these genetic sources and the non-genetic sources. And I'm interested in the non-genetic sources that drives these cell fate choices. And altogether, we can group these non-genetic sources and define a cellular state. And therefore, we think that we define, many others, an internal cellular state that affects this cell, that potentially can affect the cell fate decision process. In particular, I am interested in internal cellular states that are rhythmic. So I'm interested in looking how internal cellular rhythms affect these choices in the context of DNA damage. And today I will discuss three of them, starting with P53. P53 is a central protein in DNA damage response, and under certain conditions oscillates with 5 hours. And these oscillations have been shown to affect the cell fate choices. I'm also interested in the circadian clock that regulates many processes in your organism, but also is found at individual cell level, and it's also involved in the response to treatment. I'm also interested in the cell cycle, and here we have much longer evidence, particularly in the context of radiotherapy, how different cell cycle phases affect this treatment response. But at the end of the day, what we want is from a given treatment to understand at a single cell level, what is the cell response phenotype? How the cell, so if the cell survive or not, and also the details of that process, when it happens. As an illustration, I'm showing you one example, but it gives you a broader view of what have been the focus of within the last 15 years, I would say. At the single cell level, people have been focusing on studying the signaling dynamics that follows a treatment. And in this case, I'm showing you p53, and here many works from the lab have shown that the concentration of this protein, p53, if we follow it in time, there is a particular dynamics like oscillatory or sustained, and these dynamics drives the cell fate decisions. But we know that for oscillatory system, the way they react to a perturbation strongly depends on the state of the system at the time of treatment. So in my work, I extended this time window of observation to include the state of the cells at the time of treatment. But again, in oscillatory systems, you cannot take a single snapshot and determine a state. You need to monitor the system before that. So we extended this even further. And now we study the dynamics of internal factors in an unperturbed pre-treatment state to understand the state at the time of treatment. And then we follow proteins involved in the decision process to decode the signals in the perturbed post-treatment state. And we use all this information to understand the cell response phenotype, but to predict it. And in order to do this, we establish a four-step pipeline that involves first a design. And here we do basic cell line engineering to introduce new markers. And then we study population responses for different doses and in time. And once we have candidate conditions that we think they're interesting, we move this to the high resolution fluorescent light imaging microscope. And here we spend quite a lot of our efforts to keep cells as happy as possible and as long as possible. And this allows us then to collect multiple time points, and to then doing computational analysis, we can follow every individual cell in time and define the internal state as a moving time and a space and connect this to the outcome cell fate. This, of course, generates a lot of data that we then analyze, both doing data analysis and modeling. So we use time series analysis and modeling from toy modeling to more mechanistic modeling. And we use modeling as a strategy to understand the data and design new experiments. And we try to do this in iterative fashion. And I will show you now in the next slides an example of how we use this type of approaches to understand something about the response to chemotherapy. So I will talk about the single cell responses to chemotherapy in response to the Cisplatin, which is a widely used chemotherapeutic drug that cross-links the DNA and disrupts replication and transcription, and is used against many types of cancers, sarcoma, cancer of soft tissue, bones, and others. So as many other chemotherapeutic drugs, if you increase the dose, you have an expected response that you will basically kill more cells as you increase the doses of cisplatin. And this is what we also see. If here I'm showing you a plot, we have in the y-axis the normalized cell number of cells and for different doses of cisplatin. And we see kind of an expected response for higher doses, less cells survive. But if we look at this in time, and here we split the experiment into an untreated phase for 2 days, and then we add a different gradient, we add cisplatin in the gradient of doses. Here we see that the response depends on a time and a dose-dependent manner. And where I'm deeply interested is to understand for every of these trajectories, what are the different cell fate choices. So how many cells are dying, are resting, and how these events are being organized in time so that they all together define these population responses. And to understand those details, we need to zoom in the individual cells. So if we do that, here I'm showing you in a sketch of what is the response when we add cisplatin to the cells. We see that the cells also for cisplatin show different outcomes. In this case, we see some cells continue proliferating as if nothing had happened. Some other cells arrest, some of them by doing very early division and then arresting, others immediately, and others die through different ways. And interestingly, we see this heterogeneity even when we have a clonal population of cells that do damage at the same time with the same dose, we still see some cells going through completely different directions. So to understand how these distributions of cell fate respond, we wanted to first start with to characterize the natural, let's say the untreated condition of this, and then slightly increase the dose of cisplatin and see how these populations, the distribution between these populations shift. So if we characterize the untreated conditions, we see that a very small percentage of cells died, and then around 80% of the cells do one or more divisions. Then we have 16% of the cells that are basically arrested throughout our recordings. And as we increase the dose of cisplatin, we see something expected, that more cells died. But also, more importantly, what we see is that for all doses, we have heterogeneous cell-fed outcomes. So in all cases, we have some cells that survive and others that do not survive and take different outcomes. So for us, the main question becomes to understand what determines these individual cell-fate choices. And today, I want to focus on something that is shared among all cells that are proliferating, and that's the cell cycle. So every cell that is proliferating, you can in theory at least identify the cell cycle phase and where they are as they continue growing and divide, and what we call a proliferation behavior that will become more clear in the next slides. So to track the cell cycle on an individual cell level, we use a well-established method based on the Fuji system, in which we follow the concentration of a protein. In this case, I'm looking at geminin, and we look at the accumulation in the nuclei of a cell, and we can see it accumulates and goes down in time. And if we track this cell, we see that geminin accumulates quasi-linearly, and a division drops, then there's a silent phase, accumulates, and so on. And then we can map this silent phase and accumulation as the different cell cycle phases of these cells. And we can do this with hundreds of cells. Here I'm showing you a plot of 0 to 800 cells and 0 to almost 120 hours, that's 5 days, in which we originally have a completely desynchronized population of cells, cell cycle-wise desynchronized, and then we computationally clustered them at the time at which we add cisplatin. So these cells are at G1 at the time of cisplatin addition, these cells an S phase, these cells and the G2 cluster. And then we sort them for those cells that died. In black, I'm showing you the cells that died, and then we sort them by the time of death. So these cells died earlier than these ones. And this now allows us to ask a very simple question, which is, for example, what is the likelihood of dying if the cell receives cisplatin in G1? And from the literature, we expected this to be cisplatin to be cell cycle dependent, but what we saw when we do this careful single cell analysis is that the cell cycle seems not to affect the response to cisplatin. So then moving forward, we wanted to understand the proliferation behavior of cells and to see if this affects the responses. And this is inspired by tumor biology. And there we know, or it's an everyday practice, to look at the proliferation of a tumor to predict, to decide about the therapy treatment. And for this, we measure, or pathologists measure the proliferation index of a cell, of a tumor. And this tells you, for example, that for many drugs, if a tumor is highly proliferating, then the response would be better to certain chemotherapeutic agents. So we wanted to understand this proliferation, but at single cell level, to see if we can also test those ideas. And to measure proliferation, we can directly measure how cells are dividing, when they are divided, and so on. So here I'm showing you the proliferation behavior of untreated cells. These are 300 cells only, and we color code each division event. So here we have cells that has 1 division, 2, 3, 4, 5, and that's the division event. And then if a cell died, we mark it as black. And then we cluster them by the total number of divisions that they have within this 5 days time window. And you see a first cluster of 5, so 1, 2, 3, 4, 5 divisions, 4, 3, 2, and 1, and 0. This is arrested, and then some cells are spontaneously died. To me, this was very interesting, and it is still very surprising in a way that we always see this heterogeneous distribution of division events, in which we have cells that highly proliferate and everything in between. And we see this also in different clones. We see this in different cell lines. That means if we take one single cell out of this experiment and we let them regrow, and we take them from this group, for example, we still recover such a diversity. So, and I found it very interesting, because I always had in mind before starting these single cell studies that as a reference, this doubling time of cells, and I expected somehow a more homogeneous distribution of cells. But moving forward, what we were interested is to understand if these different proliferation behaviors can also help us to understand how are the cells going to react to a treatment. So for this, we came up with a classification, what we call the proliferation index, the single cell proliferation index. And we monitor the cells initially for 2 days to classify them. So if the cells divide 1 time or less, we call them low proliferative cells. And if the cells divide 2 times or more, we call them high proliferative cells. And then we did a very simple experiment. Now we have them classified. We just add basically cisplatin to them at different doses and see if they react differently. And in this case, I'm showing you a plot where we look at the death ratio of cells for 0, low, medium, and high cisplatin. And we have 2 groups, basically the low proliferative and the high proliferative cells. And here what we see for high doses is that it doesn't really matter what type of cell you are. High doses of cisplatin will basically kill most cells. But for the other doses, we see a very consistent picture, and that's that the low proliferative cells are more sensitive to cisplatin. So the low proliferative cells, the one without dashed lines, always have a higher death ratio. And this was exactly the opposite of what we expected based on tumor literature. So then we want to understand what is then making, so what makes these low proliferative cells more sensitive to this chemotherapeutic agent cisplatin. And we came up with 2 relatively simple hypotheses. One is that the low proliferative cells, for some reason, might be receiving more damage, that the cisplatin somehow is getting into the cells more efficiently, and therefore inducing more damage and therefore inducing more death. And the other one is that the low proliferative cells, even before the damage, might have already higher levels of basal DNA damage. And therefore, when we add cisplatin to them, then they're already closer to the death threshold, let's say. So to test the first hypothesis, if the low proliferative cells have received less damage, we measure the amount of cisplatin that the cells absorb by looking at an antibody that is the informative of the cisplatin uptake. And so we measure the mean nuclear cisplatin uptake in the nucleus of low proliferative cells and high proliferative cells after treatment. Here we see basically no difference between this population. So all cells receive equal amount of, appear to receive equal amounts of cisplatin. Then we went forward with the second hypothesis, if the low proliferative cells have higher basal levels of DNA damage before the treatment. And for this, we measure DNA damage by looking at the earlier mediator of the DNA damage response pathway, that's the gamma H2AX. And we measure the total intensity in the nucleus for cells that are untreated. So this is before the treatment. And in this, we even include one more, which is the non-divided. So the cells that are arrested, the low proliferative and the high proliferative cells. And here we see a clear trend in which the lower the proliferative cell, the higher the DNA damage that the cells have. And with this, we were quite satisfied in a way, because we can find a relatively simple explanation to what we were seeing. And that is that the low proliferative cells have higher levels of DNA damage, and this makes them more sensitive to additional cisplatin-induced DNA damage. Or another way, it could be that the cells acquire randomly, all of them, DNA damage. Those who acquire higher levels of DNA damage, then they divide less. And those are the ones we classify as low proliferative cells. To these cells, already high level, we add more damage, cisplatin-induced DNA damage, and these have higher, it's more effective because cells already are pre-damaged in a way. And the opposite happens with those cells who have lower levels of DNA damage, then they can continue dividing freely. And to those, we add cisplatin, and they have an average lower amount of DNA damage. Sorry, lower death likelihood. And this we published last year, and then but it was not able, we were still not able to explain all the variability that we observe in our data. And as I mentioned at the beginning of my talk, I'm interested in other factors that are internal of the cell, that also affect the response to treatment, in particular in the context of Cisplatin, for example. So then I will now in the next slides discuss the role of the circadian clock in the context of cancer treatment. And for some of you who are not familiar with the circadian clock, it's a system that basically makes you being awake. Now I'm paying probably a lot of attention to my talk. And this is a system that has its main nuclear center in the base of your brain, in the hypothalamus, in the base of the hypothalamus, and it's called the suprachiasmatic nucleus. This nucleus receives information from the environment, the daylight signals, and then processes information and sends signals to the rest of your body, core temperature rhythms, wake-up rhythms, and hormonal rhythms. And more important, or also importantly, it sends signals to all the peripheral organs. So all your organs have a circadian clock. And within these organs, the individual cells have a clock. And this circadian clock within the individual cells control many processes of the cell, like the cell cycles, apoptosis, and DNA repair, which are very important for the context of my talk today. And this is an exploding, I would say, research field in which people try to use circadian concepts or the circadian system to understand the relevance in the context of treatment. In different types of treatments, I'm interested in the treatment in cancer, and in particular interested in a specific question within chronotherapy, which is what is the optimal time of the day for treatment? And this is very simple. This is, for example, nowadays patients, we don't have a preferential time of the day to say if they should go in the early morning or in the late afternoon for a particular treatment. We have many reasons to believe that's an important variable when deciding the treatment, but nowadays we don't consider that. And I am interested in understanding the relevance of this question at the individual cell level. So we introduce, so we look at the individual circadian clock to understand if the cells treating the different times of the internal clock, so it's an internal 24-hour rhythm, react somehow differently to treatment at different times of the day, sorry. For this, we monitor the circadian clock by introducing a fluorescent reporter, also like in the cell cycle case. And here I'm showing you a U2S, or osteosarcoma cells, tumor cells, happily growing in a way for almost 5 days. And we have a nuclear circadian clock reporter. And this allows us to track the individual oscillations. And by understanding the distance between the peaks, we can separate different times of the day of these cells. We can map the time of the day of these cells. And then we can ask this question, if there's a particular time of the day, if for the cells to survive more or less. I'm more interested not in whole survival, but in cell-fate choices. So we can then, as in this sketch, put all the traces that we have of our desynchronized population of cells, then sort them by the first peak, and then damage them at a particular time, and then extract the phase and correlate basically what is the internal time with the outcome cell fate responses. But this is not so simple, unfortunately. We cannot just isolate the circadian clock from other systems, particularly from other systems that also have it around 24-hour rhythm. And it is known that the circadian clock and the cell cycle are coupled with each other and share a similar period, which makes the whole separation and understanding of the contribution of the circadian clock more complicated. So, and what do we know about this? So there's many pioneering and very exciting work from the Felix Neff lab in Switzerland, looking at this connection between the circadian clock and the cell cycle. One of the early observations is that when they measure the circadian clock signal of a single cell, and they plot it here, this oscillation, and then they mark the peaks in blue, and then they mark the division events of these cells, they saw that there was always a fixed distance between the division and a reference point, like the peak of the circadian oscillation. That means that these two events seems to be locked with each other. Other type of experiments, a bit more recently, they changed the period of one of the oscillators and of one of the rhythms, and the other rhythms appear to follow it. So here we are I'm showing you results. These are all from the Felix Neff lab, in which they change the temperature of the experiments, and then both periods accommodate, and they both still in a one-to-one, so they share the same period. And as I told you before, I'm interested not in this a period, but more in the specific timing. So what is the time of the day? And this in oscillators, you can map this as a phase. So in the next slide, I will tell you how we quantify the phase, which would be, for example, the phase would be the phase would be, for example, this peak. We can take a reference point as a phase. I'm interested in the phase difference. So I'm interested in the distance between these two oscillators. And for this, we'll move to what we call a phase locking. So these systems are phase locked. That means that they are moving together. Like my two hands here, or better in the screen, you see these two pendulums. They are exactly in phase. That means the phase difference is 0. These systems can still be following each other, but be completely out. Sorry, I was going ahead of myself. So, and then you can plot this. What is the circadian phase normalized and the cell cycle phase normalized, and see that one phase of the other, one phase from one oscillator corresponds to the other. You can see how one corresponds to the other, and plotted in these phase maps. But the system can be still synchronized, but slightly out of phase, so without a phase difference 0. And here what we see is that when the systems are still at the same period, but following complete different, so being out of phase, we can see that this line, this segment breaks in two pieces, but it's still, the relationship is fixed. Now, if we have two oscillators that they are completely isolated from each other in a way, and there's no phase locking, what we see is that the phase relationship is not stable, and we have all possible combinations in theory. And this, now we wanted to see, looking at our cells, introducing both the circadian clock and the cell cycle, can we now understand what is the phase relationship between these two oscillators? And surprisingly or not, what we see is that the circadian clock and the cell cycle have a strong phase locking. And this has been observed before, but we did that in much higher numbers of cells now, that we can follow in each of them the circadian clock and the cell cycle. And then we plot the phase, circadian clock phase, timing of division normalized, and we see strong signatures of a one-to-one locking with that they are out of phase. So what I'm interested is in the context of this coupling, what is the role of the coupling between the circadian clock and the cycle in cell growth and the response to treatment. And here the idea is that the circadian clock controls many processes involving the cell fate decision, the same for the cell cycle. And the fact they are tightly coupled, as you see here in this untreated condition, might indicate a certain time organization of these events for the cell. And in order to understand the relevance, we wanted to start perturbing. And for this, we are now exploring different strategies to disrupt internally this coupling within each individual cell and see how the response to different drugs or the overall growth changes when we modulate this coupling. And here I'm showing you two conditions and how we are achieving this so far, in which we can start in disrupting this locking until the point where we believe we are completely destroying it, or we have very little signature of locking. And we do this with cells that are still dividing and still circadianly oscillating. So we're not stopping the rims of one or the other, but we are modifying this internal locking. All right, but as I show you at the beginning of my talk, these rhythms are not only two things that are oscillating and relevant for the cell fate decision process, but there are also multiple other systems that are involved, like the p53 that I showed you earlier. So now what we did is we started to the silence that already have two different markers. We added one third marker of the circadian clock, and I'm showing you here a recording of cells that have these three markers. And these cells are, again, unsynchronized, and they're not stimulated with any damage. They're just, in a way, freely oscillating in constant conditions. And here we overlay the three channels. That's why you see this colorful picture. And you really see the cells doing the same transition of colors, or cells are following the same trajectory of colors. And these are, so the cells are, it seems to be that are naturally quite asynchronized in these rhythms. But then we follow each individual of each of these cells, and we try to understand what is the relationship between these rhythms. And here we are still expanding and trying the different ideas of how to characterize this variability, and more importantly, what is the relevance for treatment. One way of doing that is to plot the cell state of the cells. So, use a state space representation of the cells. And to do that, we remove time, and we plot in every axis here one of the variables that we're measuring, and we put a dot color-coded by the amount of the relative amount of the different components. And every dot on this plot indicates what's the level of decay clock, p53, and cell cycle for a particular single cell as it moves in time. But we remove them, so every point here is a different cellular state of a single cell. Interestingly, we take an image each half an hour in this recording in particular. But the time that the cells spend in different places of this space is significantly different. So you will see here, for example, that cells seem to move out very quickly out of this part, but they are in this area, they remain quite a lot. And from here, there's many things to, especially inspired from theory, many things you can investigate, and we are testing it slowly, one by one. And one of them, to me, very attractive, is to understand if the cells follow certain surface. So from here, you see, for example, that cells are not going all around. There seems to be somehow in a surface where it's called also manifold. If we look at 3 different cells, I'm adding 3 more cells in the bottom of this plot and plotting the exact same. We see that the cells somehow stay in this surface of manifold. And with this, I would like to start to close up my talk, but this is the kind of ideas we are moving towards in the future. So we are trying to add more and more live reporters that we can follow in individual cells simultaneously and understand the relationship between the different pathways as they move in time. And ideally also identify these manifolds in which the cells likes to move. And from the translational part, we would like to understand if there's particular combinatorial, so particular areas or places within this type of plots that the cells are more sensitive to a particular cell fate outcome. And other thing that I'm also very interested to pursue in the next years is to understand or to connect 2 different types of experiments. One is, the bottom one is the one that I show you today a few data, which is this low dimensional. That means we measure maybe 3 or 4 proteins together, but we do this with a high temporal resolution. And we can separate untreated to treated, distinguish the outcome cell fates, but we always have a very low dimensionality. So we can measure very few proteins. The other one is if we do multiplex imaging, we can measure multiple proteins, I don't know, 30, 50 proteins together, but we can do this discretely at different time points, and we lose the cell identity, right? And I'm very interested in connecting these 2 experiments. So in principle, we can do this in both, in the exact same conditions, same cells, same treatment, and then try to connect this high dimensional, but with low temperature resolution, with our low dimensional and high temperature resolution measurements. And this is a project for the next years. And with this, I would like to thank you and finish. First, these are the lab members of my lab for this summer. Not for this summer, but a picture taken this summer. And then these are the collaborators. The ones involved in the first part of the project is Khalid Dahab, Jacob Stewart-Orstan, Simone Redder, and Niels Blutgen. And the rest of the collaborators are from the data that I show you later. And also, importantly, my funding agents that were also supporting the work that I show you today. And yeah, with this, I would like to finish and thank you.
Thank you, Adrián. So again, please write your questions in the chat, or you can also raise your hand and then join us and ask your question in person. And maybe I can already start. So is it possible in culture to synchronize basically the circadian clock by synchronizing the cell cycle?
So we haven't managed to do that. So there is, when we do any perturbation to the cells, we always reset indirectly the circadian clock, or we reset the circadian clock. So even the 2 degrees media change will reset your clock. So I have not managed, I'm not aware of other experiment that can isolate a resetting of the cell cycle that drives a resetting of the clock. We will always see a resetting of the clock, or we try our best to avoid it because we want to have a wide diverse of phases, but we always see that very little perturbations already have a phase shifting effect.
Okay. Thank you.
Yeah.
Then Alexander, you have a question? Feel free to join.
Thank you very much for a very interesting talk. So I have a question. If, for example, you mentioned that there are some low proliferating subpopulation of cells and so on. So my question is, if you look on some biomarkers from the transcriptome, is it possible to distinguish these cell subpopulations? So I mean, have you thought about a single cell RNA sequencing?
So I'm very interested. So I have not done such experiment. I'm aware that you can. There's tools nowadays by looking at expression data. We can, or probably since many years, that we can separate cell cycle or distinguish cells at the cell cycle phase. They might be at the time in which you measure them, and the same more recently with the circadian clock. So there's ways that we can, with a single snapshot measurement, basically extract information about the specific cell cycle and circadian clock phase. I haven't done that in our lab. Yeah.
Okay, thank you.
Thank you. Then Michael Rutz, who will give the presentation in 2 weeks, also has a question. So he says, great talk, and thank you. How are cellular behaviors, so fate choices, proliferation rate, and this chemotherapy sensitivity affected by environmental signals such as different growth media and cell densities?
Yes, so this is the details of many of our experiments. We spend a lot of time testing. So a short question is, everything seems to depend also on density and growth conditions, especially specifically what we are looking at. So we are looking at very subtle variations of growth or timing of death. And we spend quite a lot of time looking at conditions that we can trust in a way, that we see, for example, a dose response curve for different growth media conditions. And we try to be in an area where the system is not very sensitive to our variability from experiment to experiment. So the answer is yes, it depends, and we try to control for this, and especially in our case because we're interested in also in arrest phenotype or changes in proliferation. Yeah, so we can discuss. We can go very much into details if you want, if we want to follow up this afterwards. But we try to define both densities and growth conditions so that the phenotype that we're interested in is not affected within the time of observation of our experiment. This is our ultimate goal. Yeah. This is kind of an answer.
Thank you. And maybe since you mentioned the arrest, I was wondering, is it actually possible within your experiments also to investigate the arrest phenotype more? Like for how long they stay arrested, what determines how they re-enter or how they exit the arrest phase? Is that possible?
Yeah, so we avoid, I did it, I think in one slide, but I avoid a long-term arrest definition in our studies. So that would be a senescent phenotype. So we cannot define the permanently arrested cell. We just define a cell that is arrested within our time of observation. And recent works from the lab actually have also shown that if you monitor the cells even longer, some of the cells re-enter the cell cycle. So cells can be arrested for 5 days, some of them, and then come back. So we cannot certain the arrest that the cells are arrested. We said they are arrested for the time that we look at them. And related to the previous question, what I meant is that we try to avoid that the cells arrest because of, for example, confluency problems or confluency factors, or because they run out of nutrients, or because, or that they do not, So this is the way we try to, I mentioned that we want to avoid, or we carefully try to control for other factors that can induce arrest, because this is something we are interested into, but we define arrest within the time we can define it, basically. And this we put very clearly in our publications also.
Thank you. And maybe one more question.
Mm-hmm.
So you do this with cancer cells. Is
Mm-hmm.
there somehow the option to also have basically a normal primary cells that you follow with similar technologies and then try to compare?
Yeah, so we are moving our efforts to do so. In principle, you have reporters that are virus or that are plasmid that we can insert into the cells. And this depends on the specific cells, how if we can basically put virus into them. And in some cases, this looks more promising, in some other cases does not. So to me, something that I really want to do in the next years is some slide that I have here in the back. Something that we are also aiming is to do this in more realistic models, using 3D models, using cell line spheroid models or patient-derived organoids. And the idea there is to introduce some of these markers. And how efficient can we do this, and how general is this is a little bit an open question. But there's already antecedents people, you can infect these cells and put reporters on them. We will be very happy already to have nuclear markers on this, so that we can assess proliferation features, migration, and this, and then the phenotypes arrest and proliferate. We can distinguish by following and tracking these cells in time. And we are aiming to do this in a particular subtype of breast cancer, which is triple negative breast cancer. But I have we are not there yet.
Mm-hmm, thank you. And then Heiner Schrewe had a question. Can you?
Hi, good morning.
Hi.
I have a question concerning the status of highly proliferating. So do these cells switch to a low proliferating state very often?
So let's go maybe to our classification. So what we see is that they switch. So for the time that maybe I can formulate it differently. I believe the cells will, some of the cells will grow. Let's say the high proliferative cells. Some of them might then acquire DNA damage as they divide, and then will then go to a low proliferative cell. And I say that because we observe such a diversity in clones, and we can regenerate it. So, and when we, through different passages and different clones and different cell lines. So we usually see these proportions between the proliferation behaviors. Yeah, so switch, yes, that would be my guess. But I have not followed an individual cell out of each of these populations, and then run the following experiments. Would be again monitor for 5 days and then characterize with the cells from the same experiment. I have done this with different generations of the same cell line.
Because it would also depend on the response of the cell, yeah, so to the drug.
Yeah.
Okay, thank you.
Then we have one more question in the chat. Maybe you can also open it by Seyoung Kim Han. And the question relates to a question regarding the understanding on whether now the clock doesn't seem to, or you try to identify whether the circadian clock maximizes the effect on drug treatment, like a time point in this. But earlier you showed that the cell cycle phase does not seem to have an effect. And since cell cycle phase and circadian clock are locked, then the question arises whether it is to be expected that the circadian clock would have an effect. Can you comment on this and help to clarify it?
So we are testing multiple drugs with the circadian clock, and it's not only the feature of if it affects so that the fact that the cell cycle itself does not affect let me follow up the logic. So that the circadian clock the cell cycle does not affect the response to cisplatin, and because it's locked to the circadian clock, this in consequence should not affect the response. It might be true for cisplatin, and we are exploring different drugs for this on different cell lines. So I would not conclude that from this single experiment. I would then maybe this is indicative for this cell line with cisplatin, and we are exploring other drugs, and people have shown for some specific drugs that we already have as time of the day sensitivity. I'm interested in chemotherapeutic drugs, and there we are far yet from reaching that conclusion. But for far from the experiments we have done with cisplatin and U2S cells, we don't see any strong modulation for cisplatin. So for cisplatin response in U2S, but this is still, I would say, we are still working on that. I would not conclude that. I understand the logic and we, yeah.
Mm-hmm. Thank you.
So it's interesting. Yeah.
Yep. Thank you very much. So yes, and there's also a thanks for your answer in the chat. Okay, are there more questions? Otherwise, I think we are also already approaching the end of the session.
Cool.
Then we would like to thank Adrián again for this very interesting talk and the super interactive discussion. And we are looking forward to welcoming you all back in 2 weeks, so the 19th of November. And yeah, enjoy the rest of your Friday and then have a nice weekend, everyone. Bye.
Bye. Bye.